Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
Today we are going to discuss about the cell functioning. So cell function, all of you know. So under the measurement of cell characteristics, the one of the major or significant characteristic is a cell function. Because imagine you have a cell and living cell, and if it is not functioning, then what is the use? So we need to check whether the cell is functioning or not, right? So that is the major goal. So are the cells performing the function for which it has to be destined, or it has been destined? That means for a particular liver cells, whether they are doing that particular task, whether the pancreatic cells are able to synthesize the insulin, or helping to able to synthesize insulin, or whether the kidney cells like nephrons are doing its filtration properly, whether they are filtering the blood properly or not, whether the reabsorption is happening in the nephrons or not. All these things are what the cell functions. So we need to check whether these cells are doing their function or not before we use it in vivo or in the humans. So in tissue engineering, cells are chosen based on the tissue that is targeted and the function that has to elicit. That means that has to target. That means you have to choose the cell in the tissue engineering. For kidney cells, obviously I will go for the kidney cells for kidney functioning or filtration. I'll go for the kidney cells, I'll go for the nephrons, then I'll check its function. So it's about the cell and its function. Okay, for example, in case of the bone regeneration, mineralization is the primary function that is aimed for. That is the the function that has to elicit, that has to take it out from the cells. So hepatocytes should perform metabolic activity for liver, right? That is the function of the hepatocytes, okay, because that has to involve in the metabolic activity. And then pancreas should synthesize hormones like insulin in their active functional form and so on. So there are many advantages, the examples. Okay, so to determine the proper functioning, specific assays should be designed. So if you want to identify whether the cells are functioning or not, or their cell viability, sometimes what happens, cell will be living but they are not functional, that is also plays a that's why the cell functioning is very important. In order to get to know whether the cells are functioning or not, we should design some assays that should be designed. For bone regeneration, the alkaline phosphatase marker quantification is there that would provide a phenotype of the osteoblast cells. Then we will come to know that the mineralization is taking place or not. So, and markers such as osteopontin and osteonectin and bone sialoprotein, these all three things, they will provide the mineralization potential of the osteoblast cells. So they also here the meaning is to say these all things are coming support to this particular process, and finally they will provide a mineralization potential for the osteoblast cells. So another example, liver we can take, that is the principal organ for urea biosynthesis, and hence distribution of albumin, urea and ammonia by the cells would provide valuable information on the phenotype and activity of the cells cultured. That means the cells and its phenotypes, that means they should do their function. In order to do their function, we cannot directly take liver and check a liver, something like that. Indirect approach always is better option. So we know that the liver cells is a principal organ for urea biosynthesis. That means if liver is functioning properly, urea biosynthesis will be there. So, and also you can estimate the albumin, urea and ammonia. These three things you can use as an indirect approach. If the albumin is there, if the urea is there, and if the ammonia is there before and after the introduction of this thing, if there is any difference, then we can say that the cells are functioning. So before you implant the cells inside the liver, you check the estimation of the albumin, urea and ammonia should be done and kept. And then after implanting the cells, again you take out the assay, again you do the design the assays in such a way that you are going to estimate again albumin, urea and ammonia. Now you compare both the things. If there is a difference, then they can say that the non-functional or the damaged cells have been replaced by the functional cells, because whatever the cells you have implanted in the liver are functioning now. Understanding? So that was about the cell functioning. In the similar way we are having for many organs. Okay, same thing goes for neuronal cells. Okay, where nervous system we uses the nerve cells, where the nerve cells gives you the signal from one nerve to the another nerve. A signal transfer will be there, that we will give you the communications. Okay, so all these things requires that particular, you need to identify a particular some chemical which is being produced, and then for example in like how we estimated albumin in liver, so that is a sign that the particular the cells are doing their tasks or the function which is destined for, or which is prepared for, or which we have used for. So in order to use it in vivo, that means inside the human being, first we need to check for whatever the cells you are using. It can be the kidney cells, liver cells, pancreatic cells, heart cells, even brain cells, whatever the cells you are talking about. Those cells need to be studied whether they are functioning or not. And here most of the time you will be using indirect approaches. For example, you have a reactant and then you have a product, right? So now in order to these reactants, imagine the cells are there, and then there is some cells are there, and cells are also one of the reactant. Now imagine this particular reactant is responsible for this product. Okay, albumin is a product. Now I need hepatocyte cells for that. So when I'm using the hepatocyte cells, okay, which are non-functional, why we use tissue engineering? In order to replace non-functional cells with the functional cells. Now the first thing, I will take a liver cells where the albumin production is not proper, urea production is not proper, ammonia production is not proper. So what I will do, I will check first, then I will keep it as it is. Then I will grow the cells which are healthy cells. Then I will check again, same thing, reactant, product. So now I will measure the albumin which has been produced with the healthy cells, or which then I can see the significant difference in the albumin concentration which has been biosynthesized, urea concentration which has been biosynthesized, or ammonia concentration which has been biosynthesized. Now these differences will clearly tell me that the cells are good to go. That means you can use these cells for your tissue engineering. That's what I wanted to explain, and that's it. The next topic we are having is the sixth major topic, that is extracellular matrix. And in this topic we have studied about three different things. It can be asked for one big question for 10 marks or a separate question for 5 marks. They can also combine two properties, characteristics, and ask for 6 marks, 7 marks also. So what was the topic? Fifth topic, measurement of cell characteristics. In that 4 subtopics we discussed: introduction, cell number and viability. We also discussed about the cell motility and migration, and finally under this only we discussed about 4 different assays like Boyden filter, transposition, transmigration assay, Zigmart chamber, Dunn chamber, and agarose assay. And finally we discussed about cell function. So always remember, whenever the question comes, it depending on the marks you write the answer, you frame the answer. You simply start from the cell characteristic or blah blah blah. Don't write. If they are asking, they'll write about the function of the cell. You start from here. Give the introduction, 2 to 3 lines. Will have 2 marks, and then cell function will have 3 or 4 marks. 5 marks is done. Or if they are asking combining these chambers, then you see separately. If they are asking separately, you write. Or else if they are asking cell motility and migration, write all these 4 different assays, and definitely all will get you 1 mark each, and introduction or a diagram 1 mark. Okay, so that's it about the 5th topic. Next topic we are having extracellular matrix, that we will discuss in the next class.